Hi, everyone. So today we're going to go ahead and review cell structure and function. So in this unit, we are going to talk about the cell structure as well as its function. So the structure is how it's built and then how it works, how it's built and how it works. All right, cellular level of organization. The detailed study of the cell began in 1830s. It was a unifying concept in biology. Basically originated from the work of biologists Schleiden and Schwann in 1838 to 1839. And what they developed was a theory known as the cell theory. You need to know this. So you need to know the cell theory. So I'm going to go ahead and highlight or mark it up. Make sure that you know this, okay? So the cell theory states that all organisms are composed of cells, that all cells come from only pre-existing cells, and that cells are the smallest structural and functional unit of organisms. So anything that is alive that contains a cell states that all organisms are composed of cells, once again. All cells come from a pre-existing cell, so cells just don't arise spontaneously, and cells are the smallest structural and functional unit of organisms. Here's a pretty cool video. I would suggest that you guys watch it. It is called The Wacky History of the Cell Theory. It is a TED Ed video, and it does a really good job of explaining the cell theory. So please take your time and watch this video. All right, these are the learning objectives for this subsection. So in this subsection, we're going to talk about cell size. So we are going to talk about the effect of surface area to volume ratios on the exchange of materials between cells and organisms in the environment. So let's go ahead. You guys can read these on their own. This here comes from the AP curriculum, and this is what the AP curriculum wants us to talk about. So in this next subunit, we're going to talk about surface to area. Okay. All right, cell size. Cells range in size from 1 millimeter down to 1 micrometer. So they're pretty small. Cells need a large surface area of plasma membrane to adequately exchange materials. So the surface area to volume ratio requires that cells be small. Cells need to be small. Large cell surface areas relative to volume decreases, whereas a small cell's larger surface area to volume ratios is an advantage for exchanging molecules. All right, so let's take, for example, like carbon dioxide and oxygen exchange. So when we look at CO2 and O2 exchange, we know that the cell is small in size, okay? So we know that molecules go into and out of the cell. So if we look at CO2, it goes in and it goes out, okay? So if a cell was much larger, as what we're talking about, so if I see a cell that's this big, so to speak, we can tell that it's going to take a lot longer for the materials to go into as well as go out of a cell, okay? So cells that are smaller tend to have a larger surface area volume exchange for that. And if you recall, what is what are the cells made up of the inside? So they're made up of cytoplasm. So they're made up of cytoplasm. So if our cells are like really big, okay, we would be like jelly blobs, blah, 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 okay? We'd be like a big blob because every time a cell makes a copy of itself, it's made with a jelly-like fluid. So instead, we have these smaller cells that allow for materials to exchange. Okay, here's an example as we see here of surface to volume ratio. Okay, so we're looking at these 3 different cubes. Okay, and as we can see, we have one that is 4 centimeters, one that is 2 centimeters, and one is that is 1 centimeter cube. So we have one big one, 8 2-centimeter cubes, and 64. These here, 64 1 centimeter cubes, are the same as this one big cube. So if we look at surface area to volume ratio, this here has a lot more surface area as opposed to this one. All right, here are some videos I'd like for you guys, same thing, to take a look at. Why are cells small? These two videos do an excellent job at showing you guys some real life examples of the purpose as to why cells need to be small, as to why they need to be small. And I will be showing you guys too also a lab. So I will be doing a teacher hands-on lab in which you'll be able to see this as well. All right, let's go on to our next topic. All right, prokaryotic cells. So now we're going to go ahead and talk about the difference between prokaryotic cells and eukaryotic cells. So this is something that you should know, I'm hoping, and you should be reviewed. So if it's not, then you are going to have to kind of recall some of this stuff. So let's go ahead and look. Anytime we see pro means before or no, okay? Karyote means our nucleus, and then obviously your cells. So prokaryotic cells lack a membrane-bound nucleus. They lack a membrane-bound nucleus. Structurally, they're smaller and simpler than eukaryotic cells, which have a nucleus. Prokaryotic cells are placed in two taxonomic domains. They include bacteria and archaea bacteria. Remember, archaea live in extreme habitats. So the domains are structurally similar, but biochemically they're different. What you need to know about prokaryotic cells is that they don't have a nucleus, and they are known as bacteria cells, All right, the structure of prokaryotic cells, they have three different types of shapes. So prokaryotic cells come in three different types of shapes. They're spherical, they're rod shaped, or they're spiral. So spherical, rod shape, or they're spiral. The cell envelope includes a plasma membrane. So the outside, they also have a plasma membrane. Same thing, it's made up of a lipid bilayer with embedded and peripheral proteins to help form these internal pouches known as mesosomes, okay? Prokaryotic cells also have a cell wall, and that cell wall is used to maintain the shape of the cell, and it strengthens the peptoglycan. And it also has this structure called this glycocalyx. And basically what that is, it's a layer of a polysaccharide. So if we recall what polysaccharide is, polysaccharides break down to your sugars on the outside of the cell wall. And it's well organized, and it's resistant to removal of the capsule. So these are the shapes of what bacteria look like. So these are the different shapes. Here's another structure. Here's our structure of our prokaryotic cell. Notice here, as we were to see these, it has our nucleoid region. So it does contain DNA, but there is no nucleus. There's no nucleus. It's kind of just free floating. It also has ribosomes. So bacteria all have ribosomes. It has this finger-like structures on the outside, which are called fimbriae, which helps the bacterial cell move around. It has a plasma membrane, also known as the cell membrane. It also contains our cytoplasm. Okay, our prokaryotic cells contain cytoplasm, just like our regular cells. And the cytoplasm is a semi-fluid solution. It's bonded by the plasma membrane, which contains water, inorganic and organic molecules. So inorganic could be like carbon dioxide and oxygen, and it also contains enzymes, okay? Prokaryotic cells, like we said, also have a nucleoid region, and it's a region that contains the single circular DNA molecule, and it also has plasmids. You're going to have to know this term as well, plasmids. Okay, so this is something that is new. So plasmids are small accessory rings of DNA. It's rings of DNA. When we get into biotechnology, we're going to revisit what a plasmid is. But for right now, you need to know that's a ring of DNA, which is found in a prokaryotic cell. Okay. We also know that prokaryotic cells have appendages that allow for movement. So there's three types of appendages that we look at. They have a flagella, which is tail-like, and it provides motility. It allows for these organisms to move. It has fimbriae. Fimbriae are these small bristle-like fibers that sprout from the cell surface. And it also has an area which is known as a conjugation pili. And basically, it's a rigid tubular structure which is used to pass DNA from one cell to another. So it's basically the structure that allows prokaryotic cells to have sex, so to speak. I know it sounds kind of gross, but these organisms have to obviously exchange DNA information with each other as well, okay? All right, I'm going to go back just to show you once again what the fimbriae look like, So fimbriae, here are the fimbriae structures. These are these little hair-like structures as you see here. So all those little hair-like structures are called fimbriae. All right, here's some other pictures. We have cilia. Cilia is also kind of like known as fimbriae. And then here's our flagella. Here's the flagella, as you see. All right, here's another video on our amoeba sisters. Same thing. I suggest you'd watch it. It does help out a lot, especially because it gives you some mnemonics and some things to remember the difference between a prokaryotic cell and a eukaryotic cell, which you will have to know about prokaryotic and eukaryotic cells. All right, so let's talk about our eukaryotic cells. Our eukaryotic cells contain membrane-bound nucleus that houses the DNA. They have specialized organelles, and they also have a plasma membrane or cell membrane. And these are much larger than our prokaryotic cells. So what's the difference between a eukaryotic and a prokaryotic? So remember, a eukaryotic cell has a membrane-bound nucleus, so it has a nucleus, it has organelles, and it has a plasma membrane, okay? Whereas a prokaryotic cell has no nucleus and no organelles. All right, how did these organelles come about? So the origin organelles comes about through this process called the endosymbiotic theory. And what this states is that one time there's this original prokaryotic cell, okay, or this ancient cell. The cell gains a nucleus by the plasma membrane and surrounding the DNA with a double membrane. So as we could see here, over time, it gains a nucleus, okay? The nucleus allows specific functions to be assigned, freeing up cellular resources for other work. The cell then gains an endomembrane system, okay? So endomembrane systems such as the Golgi and the ER, and that allows for an increased surface area for higher rates of transport of materials within the cell. The cell gains a mitochondria, and because of that, it's allowed to metabolize sugar, okay? Or it's allowed to make energy. And some cells gained a chloroplast. The gaining of the chloroplast allowed them to not only make energy, but also allowed them to produce sugar. So over time, we see how this cell evolves into a cell that can kind of function on its own. Okay. All right, go ahead and take a look at this video. This is just to give you a basics on the endosymbiotic theory. We will be doing a little bit more with it later. So I know I just gave you basics, but we're only going to talk about the basics for now, okay? All right, eukaryotic cells. Let's get into the meat and potatoes of this. Okay, let's look at the organelles. We have learned about organelles forever. You probably learned them about organelles in 7th grade, and then you learned about them in regular bio. Now we're going to learn about them again. So eukaryotic cells are compartmentalized. You got to know this term as well, compartmentalized. And if I look at the word compartmentalized, I see the word compartment, okay? They contain small structures called organelles, and each organelle performs a specific function, which isolates reactions from others. So every organelle works on its own. Now there's two classes of organelles. The endomembrane system, so make sure that you know this term, endomembrane system. So endo means inside, and then obviously membrane, so it's going to be inside the membrane. And then we also have the energy-related organelles, okay? So endomembrane system are organelles that communicate with one another, so they work together. They work together via membrane channels and these small vesicles. The energy-related organelles are the mitochondria and the chloroplast. And what they are is they're basically independent and self-sufficient. So mitochondria work on their own. All right, here are the learning objectives. for this unit as placed due from the College Board. So in this section, we're going to be able to describe the structure and or the function of our subcellular components and organelles. We're going to focus on the ribosomes, the ER, and the Golgi for right now. All right, let's take a look at the nucleus and the ribosomes. The nucleus, as we know, is the command center of the cell. It's usually near the center. So make sure you can say that, nucleus, right? One more time, nucleus. Okay, good. It's separated from the cytoplasm by a nuclear envelope. It consists of a double layer membrane. Okay, notice here, double layer. So the outside of the nucleus is a double layer. So it's made up of, you guessed it, a phospholipid bilayer. So it has that. And sorry, my drawings aren't the greatest, but I'm hoping you guys can tell. So it has a double layer. Okay. It also has nuclear pores, and these pores permit exchange between the nucleoplasm and the cytoplasm. So there's pores that allow for things to enter and exit the nucleus as well. Okay. It contains chromatin in semi-fluid nucleoplasm. So chromatin is another term you got to know, chromatin. And chromatin contains DNA of genes and proteins. It condenses to form chromosomes. It forms the chromosomes. Okay? Chromosomes are formed during cell division. Also within the nucleus is this structure called the nucleolus. And the nucleolus makes ribosomes. So make sure you know that. The nucleolus makes ribosomes. Nucleolus makes ribosomes. So this is the anatomy of the nucleus as we see. Here's our nucleus. In the middle is the nucleolus. Like we said, this is where the ribosomes are made. We have these small little pores. This is where our RNA would leave. And as we can see here, the membrane is a phospholipid bilayer, phospholipid bilayer. All right, the ribosomes, what you need to know for the ribosomes. The ribosomes are the site of protein synthesis in the cell, or where proteins are made, okay? So remember, when we're learning about all these organelles, you need to know the structure and the function. So what does it do and how is it made? What does it do and how is it made, okay? Ribosomes are composed of rRNA. rRNA stands for ribosomal RNA, and it consists of a large subunit and a small subunit. These subunits are made in the nucleolus. They may be located, so ribosomes can either be located on the ER, making it rough, or they can be free-floating in the cytoplasm. So ribosomes are either singly or they're in groups called polyribosomes. This here is what the structure of the ribosome looks like. There's a large subunit and a small subunit. And like we said, they're used to make our protein. So basically, if you recall, our mRNA strand would attach. There's three big structures located here. And later on, we'll talk about those. But this here is where we read our codons, if you recall, from our mRNA strand. Okay, so we have our ribosomes that are located here. These are on the ER, and this is going to be a rough ER, or we have some that are kind of just floating around, and those are free-floating ribosomes, okay, free-floating. Here's a function of our ribosome, as we said. So our ribosomes that are attached onto our ER, they're responsible for making proteins. So our RNA attaches to the ribosome. As it attaches to the ribosome, it forms this really long polypeptide chain. So remember, our polypeptide goes through our primary, our secondary, our tertiary, until it finally makes our quaternary structure or our last structure. All right, the endomembrane system is a series of intracellular membranes that compartmentalize the cell. So intra meaning inside of the cell. They restrict enzymatic reactions to specific compartments within the cell. The endomembrane system consists of the nuclear envelope, membranes of the ER, the Golgi apparatus, and vesicles. Vesicles are several types, and what they do is they transport materials between organelles of the system. All right, the ER, also known as the endoplasmic reticulum. It's a system of membrane channels and saccules, which are continuous with the outer membrane of the nuclear envelope. Same thing here. The membrane is made up of a phospholipid bilayer. So the ER, in essence, is basically attached to the nucleus. Okay? We have rough ER, and then we have smooth ER. Rough ER contains ribosomes, and it aids in protein anabolism, or it helps to make proteins. The purpose of the rough ER is to modify and process proteins. So it modifies and it processes the proteins, results in what are known as glycoproteins. Why? Because it adds a sugar to the protein. Next, we have the smooth ER. Smooth ER has no ribosomes, and it forms lipids, or it helps to synthesize in lipids. The site of various synthetic processes such as detoxification as well as storage, and they help to form transport vesicles. So smooth ERs help to detoxify materials as well. So smooth ER helps to detoxify. Why is that important? Well, when we start learning about different types of cells, right? Different types of cells are going to have different types of organelles in numbers or in numbers. What does that mean? So if we look at, like, you have cells in your stomach, you have cells in your large intestines, in your small intestines, you have skin cells, eye cells. All of those cells contain organelles, but they have different amounts in them depending on what the job of that cell is. So if you look at detoxification, which organ in your body detoxifies? There's two. I'm hoping that you chose the kidney and the liver. The kidney and the liver help to detoxify toxins from the system. So because the liver and the kidney help to detoxify, they are going to have large numbers or a lot of smooth ER. Why are they going to have a lot of smooth ER? Well, because smooth ER helps to detoxify. Okay. This is what the ER looks like, as we said. So here's the nucleus, and as you could see, it's kind of attached onto these pores and so forth. So here is the nucleus is right here. Here's our ER. It has ribosomes that are attached. So the attached ribosomes are made up of, or help to produce proteins. And then towards the end is going to be the smooth ER. So we have rough ER and then we have smooth ER. Both of these are made up of phospholipid bilayers, once again. The next organelle that works with the ER is called the Golgi, or the Golgi apparatus, and it consists of flattened curved saccules. It resembles the sack of hollow pancakes. And what the Golgi is responsible for doing is it helps to modify proteins and lipids. It receives a vesicle from the ER on the cis or the interface side, and it packages it into a vesicle. It prepares the protein to be shipped and packaged to go in and out of the cell within the cell, and it helps for export from the cell. So whenever we're making things such as secretion or in exocytosis. Okay, so once again, the Golgi prepares the material, and then it's going to ship it to a vesicle. The vesicle then will attach to the cell membrane so that it leaves. So let me give you an example of what that looks like in a second. All right, so here's our Golgi. So once again, here's the nucleus, here's the ER. So let's talk about protein secretion. So let's say, for example, you need to make lactase. You drank milk, right? So now that you drank milk, your body has to make the enzyme lactase to break up the sugar lactose. The information is found here in the nucleus, in our DNA. DNA then is going to be transcribed into RNA. RNA then is going to attach to a ribosome, which is found on the ER. The ribosome is going to be made. Then the ER is going to ship it into a vesicle, or this transport vesicle here, okay? Now that this protein is made, it gets shipped to the Golgi. The Golgi now is going to look at it, modify it, and it's going to package it into another vesicle. Now it's going to send this vesicle, and the vesicle is going to merge with the cell membrane, and now lactase goes outside of the cell for secretion. And now you can break up milk sugar, okay? Notice how everything is made up of phospholipid. We have the membrane of the nucleus. We have the membrane of the ER, the membrane of the Golgi, and these vesicles. Because they're made up of phospholipids, when this vesicle starts to attach to the cell membrane, it becomes part of the cell membrane because it's made up of phospholipids. All right, working on its own is the lysosome. And the lysosome is our membrane-bound vesicles, which are not found in plants. Okay, so lysosomes are not found in plants. Lysosomes are produced by the Golgi, and they contain powerful digestive enzymes. So they contain powerful digestive enzymes and are highly acidic. So if they're highly acidic, that means that they have a pH that is less than 7, right? So less than. And what the lysosomes do is they help to digest large molecules, and they help to recycle cellular resources. So when your organelles get old and they no longer function, they kind of become waste or trash products, okay? So we don't allow for trash to kind of add up inside of our cells. So we have these lysosomes that kind of go around the cell inside. They're kind of like vacuums. And what they do is they pick up anything that's old or dying, and they help to digest these molecules as well as enzymes, okay? Some genetic disorders, though, are associated with lysosomal. They're known as lysosomal storage enzymes. An example would be Tay-Sachs disease. So a person that has Tay-Sachs disease, basically what they do is they lack the digestive enzyme found in their brains, okay? And so if this enzyme isn't working, then what it cannot do is it cannot break up molecules such as fats, which are found inside of the cells. So the cell then starts to accumulate with trash. And as it starts to accumulate, it starts to get bigger and bigger and bigger. And as these cells start to accumulate with waste, right, they now start to take over the brain. So then the brain stops functioning, and then over time, the child dies. So Tay-Sachs is a very fatal disorder. So if you have it, there is no survival. It is sad, okay? This is what the lysosomes look like, as we see here. This is the lysosome engulfing a mitochondria, as we see. So this may have been like an old mitochondria that's no longer functioning. All right, so to summarize the endomembrane system, proteins are produced in the rough ER, and then lipids from the smooth ER are carried in vesicles to the Golgi. The Golgi apparatus then modifies these products and then sorts and packages them into vesicles that go to various cell distinctions. These secretory vesicles carry products to the membrane where exocytosis produces secretions. So exo means exit, right? So these proteins exit the cell and they produce these secretions. So secretions could be like hormones. Maybe they're making some enzymes. So they're secreting something. Lysosomes help to fuse with incoming vesicles, and they help to digest macromolecules. And what are the macromolecules again? Proteins, carbs, lipids, and nucleic acids. All right, good. Here's that picture once again. So let's go through it one more time. Here's our nucleus. Information from the nucleus leaves the nucleus in the form of mRNA. mRNA attaches to a ribosome, which makes a protein. The protein gets shipped along the ER into a vesicle, okay? This vesicle here now is going to attach to the Golgi. The Golgi now is going to package and sort and modifies it. So if there's a mistake, the Golgi will fix it. Then it's going to ship the protein now into another vesicle. This vesicle here now merges with the cell membrane and the protein gets secreted. Okay. If it's consuming, also known as endocytosis, is when your cell is taking something in. So here's this molecule here. Notice now it's forming a vesicle. This vesicle now brings the substances to the cell to get digested with the lysosomes and gets broken down. Okay. All right, other vesicles and vacuoles that you're responsible for knowing. One is called our peroxisomes. Peroxisomes are similar to lysosomes in that they're membrane-bound vesicles that have enclosed enzymes. However, these enzymes are synthesized by free ribosomes in the cytoplasm instead of the ER. And what they do is they are active in lipid metabolism, or for breaking down fat. They help to catalyze reactions that produce hydrogen peroxide. Hydrogen peroxide is toxic in our system, H2O2. So what it does is the enzyme catalase will break down hydrogen peroxide into water and oxygen, This will be a lab that we will be able to do later on. You will need some hydrogen peroxide, which I'm hoping a lot of you guys have at your homes. And that helps, like I said, we'll be able to see how catalase works with hydrogen peroxide to make water and oxygen. It's kind of cool. Here's a peroxisome as we see here. All right, next is the vacuoles. Vacuoles are membrane sacs that are larger than vesicles. Vacuoles store materials that occur in excess. Others are very specialized. Some organisms have what's known as a contractile vacuole. Plants usually have a typical central vacuole. So if you're a plant, your vacuole is known as a central vacuole. And the central vacuole has up to 90% volume of some of the cells. What's the purpose of the vacuole? Well, it helps to store water, nutrients, pigments, and waste products. It aids plants in the development of turgor pressure. Turgor pressure is the pressure that water exerts on the outside of the cell. So if you think of like a leaf in general, okay? If a leaf is happy, meaning that it has enough water, it's going to stand up straight. If a leaf is sad, meaning that it's lost water, it's going to be droopy. That pressure of the water inside of the cell is known as turgor pressure. And some vacuoles perform some functions performed by lysosomes and other eukaryotes. The main thing what we want to know is it helps to store water. Okay, it's our storage of water. Here's the plant cell central vacuole as we see here. And as we could see, it takes up the majority of the cell itself. Okay, our energy-related organelles. So let's go ahead and look at our energy-related organelles. First one we're going to start off with is the chloroplast. The chloroplast, notice here, is bonded by a double membrane. Double membrane meaning a phospholipid bilayer, okay? You have to know the structure of this chloroplast, okay? So double membrane. It also has a membrane, an inner membrane enfolded. It forms these disc-like thylakoids. So it forms these disc-like thylakoids, which form stacks called grana, okay? It's suspended in a semi-fluid called stroma. So stroma is the fluid found inside of the chloroplast. It's similar to cytoplasm, okay? The discs are called thylakoids, and they form stacks called grana. Chloroplasts are green due to the pigment called chlorophyll. So you got to know that, chlorophyll. It is a green photosynthetic pigment, and it's found only in the inner membranes of the chloroplast. It's found only in the inner membranes of the chloroplast. All right, chloroplasts have membranous organelles that serve as the site of photosynthesis. So chloroplast is where photosynthesis takes place. It captures light energy to drive the cellular machinery of the cell. A quick overview of photosynthesis, just so you recall, is photosynthesis helps to synthesize carbohydrates from carbon dioxide and water. Photosynthesis also makes, the chloroplasts also makes their own food using carbon dioxide as the only carbon source. Okay, so photosynthesis is a process where it makes its own food. So plants make their own food using carbon dioxide as its only carbon source. So basically, they take in air, CO2, and they convert that air into sugar or carbohydrate. It is energy poor compounds converted to energy rich compounds. It contains or it uses solar energy plus CO2, carbon dioxide plus water to give us carbohydrate and oxygen. So later on, you will have to know this. Super important. Okay, the formula for photosynthesis is cellular energy plus carbon dioxide plus water yields or gives us carbohydrate, which is sugar, plus oxygen. Only plants, algae, and certain bacteria are capable of conducting photosynthesis. Okay, so once again, photosynthesis synthesizes carbohydrates from carbon dioxide and water. So make sure you know what this means. Synthesizes carbohydrates. Synthesize means to make. So it's making sugars from CO2 and water. So here's what the structure looks like up close. So if we have the chloroplast, if I were to kind of like slice it in half, this is what it looks like. This here is the grana and our thylakoids, as we see here. So we have our thylakoid and our grana, or our grana. The stroma is the inside of the, or the fluid inside of the chloroplast. And same thing, it has an outer and an inner membrane, outer and an inner membrane. All right, now let's go ahead and talk about the mitochondria. The mitochondria, which are other energy organelle. So mitochondria is smaller than the chloroplast. It contains ribosomes and their own DNA. So mitochondria has its own ribosomes as well as its own DNA. It's surrounded by a double membrane, So same thing, mitochondria has a phospholipid bilayer. The inner membrane surrounds the matrix and its convoluted folds to form the cristae. So some words that you have to know are matrix and cristae. The matrix is the fluid inside. Same thing, it's similar to the cytoplasm, similar to the stroma, okay? But the matrix is the name of the fluid found inside of the mitochondria. And then the folds, the inner folds, are called the cristae. So like we said, the matrix is the inner semi-fluid containing respiratory enzymes to help break down carbohydrates, or it helps to break down sugar. It is involved in cellular respiration, and it produces most of the ATP that's utilized by the cell. So this is where all of our energy is made, right? All right, this here is what our mitochondria would look like. The same thing if I were to get the mitochondria and cut in half. We have the cristae where these little folds are located. So these folds right here are is known as a cristae, and the matrix is going to be the fluid found inside. These double folded membranes allow for more surface area, so for reactions to take place, and double membrane, so it's made up of the phospholipid bilayer once again. All right, here's an overview of all of the cells. So my suggestion would be to probably make your list and kind of go through these cells. It would make life easier when you're writing out your notes, okay? Kind of make your list and then go ahead and write down your notes. All right, the cytoskeleton. So let's go ahead and look at this term, the cytoskeleton. Cyto means cell, and skeleton is obviously the structure. So cytoskeleton is the skeleton of the cell. It helps to maintain the cell shape. It assists in movement of cell and organelles. It assists in the movement of cells and organelles. And there's three types of macromolecular fibers that we're going to look at when we talk about the cytoskeleton. They include actin filaments, intermediate filaments, and microtubules. They assemble and disassemble as needed. They assemble and disassemble as needed. So if you think of yourself, you're pretty squishy, right? If you were to grab yourself and kind of like just kind of like almost pinch your arm, you can feel like you're like water substance, okay? When you do that pinching, the cells kind of like float past each other. They move past each other. The cell will kind of like change its shape for a quick minute and go back to normal. So it has these filaments that kind of help it, almost like these elastic fibers. All right, actin filaments. Actin filaments are extremely thin filaments like twisted pearl necklace. And what they are is they're dense web, which are found just underneath the plasma membrane, and it helps to maintain the cell shape. It supports for microvilli in the intestinal cells. Intracellular traffic control is done here, intra meaning the inside, for moving stuff around within the cell. There's a cytoplasmic streaming, and it's a function in pseudopods of amoeboid cells. So it allows for like these amoeba to kind of like float around. It's the important component in muscle contraction. It's also important in muscle contraction. This is what the actin filaments look like. So these here are found within the cell itself. We would see these most likely in our muscle cells, Okay, so here's that filament here. All right, an intermediate filament. Intermediate in size between actin filaments and microtubules. So there's a rope-like assembly of fibrous polypeptides, right, or proteins. They vary in nature from tissue to tissue. The function is they support nuclear envelope, and then they aid in cell-to-cell junctions, like those holding skin cells together. They look something like this. Oops, I don't have the picture. Darn it. Okay, so my intermediate filaments, like let's say, for example, if I kind of like entangle my fingers together, I entwine my fingers together. When I entwine my fingers together and link them together, that's basically how your cells are linked to each other. So they have these intermediate filaments, which form what's called a cell-to-cell junction. And that's what holds your skin cells tightly together through these filaments. All right, the next one is our microtubules. Microtubules are hollow cylinders that are made up of two globular proteins called an alpha and a beta tubulin. They are spontaneous pairing of the alpha and beta tubulin molecules form these structures, which are called dimers. Dimers then arrange themselves into a tubular spiral of 13 dimers around. The assembly, under control of microtubule organizing center, it's the most important MTOC, is the centrosome. So centrosome is what holds the chromosomes together, and we see that during mitosis. It interacts with proteins, kinase, and dynein to cause the movement of the organelles. So these microtubules attach to organelles and they help to move them around. Here, as we can see, this is our microtubule operation. Okay, so the vesicles move, not the microtubules. Once again, here's our cytoskeleton. So we have the actin filament, we have the intermediate filament, and then we have the microtubules. All you need to know for these is that the aid in structure of the cytoskeleton. All right, next we have our centrioles. Centrioles, we most remember them when we're talking about mitosis, okay? They are short, hollow cylinders composed of 27 microtubules. Microtubules are arranged into 9 overlapping triplets. There's one pair per animal cell. So centrioles are only found in animal cells. They're located in the centrosome of animal cells, and they're orientated at the right angles to each other. They separate during mitosis to determine the plane of division. So centrioles basically attach to the chromosomes, and they help to move the chromosomes around. They may give rise, though, to some central basal bodies of cilia and flagella. So centrioles also aid in the development of cilia and flagella. Okay, so this is what we look like. Centrioles, they look like a stack of straws. Okay, and like what we said they do is they also make these small little fibers that attach onto the chromosome and they move them around during cell division. All right, finally, our cilia and flagella. Cilia and flagella. They are both hair-like projections from the cell surface that aid in cell movement. So the cilia and the flagella help the cells to move around. They vary different from prokaryotic flagella. The outer covering of the plasma membrane. So inside this is a cylinder of 18 microtubules arranged in 9 pairs. In the center are 2 singular microtubules. This 9+2 pattern is used by all cilia and flagella. In eukaryotes, cilia are much more shorter than the flagella. Cilia move in coordinated waves like oars, and flagella move like a propeller or a corkscrew. This is what we see here. Obviously, flagella were most notable for our sperm cells, right? So the tail is a flagella, which aids in movement of the cell, right? Purpose of the sperm cell is to deliver its DNA to the egg cell, and it needs to move. So the way it moves is through the flagella. All right, here's a video here on the cytoskeleton structure to kind of give you an idea of what these fibers look like. All right, that is all for today. I'm hoping that some of this was a review. Please make sure to review each of your terms and the new terminology that you use. And I hope you guys have a great day.